Hello everyone, today we're going to be doing a gram staining tutorial. First we're going to label our slide with a positive and negative control, and then we're going to have a space for our unknown. We're going to go ahead and take our loop, we're going to go put it in the Bunsen burner to sterilize it until it is glowing red hot. As you can see right here, I'm going to draw that on there. Then we're going to go ahead and let that cool for 20 seconds. As we take that out of the flame, we're going to dip it into our deionized water. Then once we dip that into the deionized water, we're going to have a little bit of water on the loop. And then we're going to go ahead and take that water and put that in our little circle that we drew out onto our slide. For the sake of time, I just added that to all of them. And we're just adding this water so our bacteria has something to stick onto, okay? It's going to also come into play when we do heat fixing later. So our next step, we're going to take our needle and we're going to put that in the Bunsen burner flame to let that get red hot. And then once we take that off, we're going to let it cool off for 20 seconds so that we don't kill our bacteria that we are going to be obtaining from our cultures. So then we're going to go ahead and take one of our slant culture tubes. So this one's going to be for our gram-positive. We're going to go ahead and imagine we're uncapping that, putting our sterile needle inside of the tube, getting a little bit of that bacteria and placing it into the water until it is a little bit cloudy. And then I'm just going to go ahead and put the needle into the Bunsen burner flame and let that get red hot to sterilize it again before I go into a different tube. And then I'm going to go ahead and speed past these next few tubes. It's going to be the same procedure for these. So now we should have a slide that has a couple of circles that look cloudy from the bacteria. And then our next step is going to be heat fixing the bacteria to the slide. So we're going to go ahead and clip that with a clothespin so that we're not holding the glass as it's getting hot in the Bunsen burner. We're just going to do some P shapes over the very tip of the flame just to dry out the water that we added before and make that bacteria, make sure that it's dead. Now let's get into our primary stain with crystal violet. So we're going to dip into the stain and just flood that whole slide. And this is going to just stain the entire slide. We're going to go ahead and rinse that with the deionized water. And we're going to see that we are going to get a purple stain on all of our circles. Now our next step is going to be to apply mordant. In this case, we're going to be using iodine, and this is just going to help the gram-positive bacteria hold on to the crystal violet rather than the gram-negative bacteria. So this is going to be making sure that our gram-positive is going to be purple. Meanwhile, our other ones should not be purple. So we're going to go ahead and put that all over the slide, and you're going to see it's going to be still purple once we rinse that all off. And the reason why all of our samples are still purple is because we have not decolorized, which is going to be our next step. So the decolorization step is what really allows us to get the pink look of the gram-negative bacteria. The decolorization step is one of the most important steps in the gram staining technique because it's how we know that our gram-negative is actually gram-negative. So we're going to be putting ethanol on the slide for only about 15 seconds until the liquid coming off is going to be clear. And we don't want to under decolorize or over decolorize because that can mess with our results. So we only want to be doing this until our gram-negative is no longer purple. And because we see that our gram-negative is not purple and it is now clear, we know that we were able to decolorize correctly with our ethanol. So now we're going to go ahead and do our counter stain with safranin, and this is how we're going to actually get the pink color for our gram-negative bacteria. So we're going to go ahead and take the dropper and place that into the safranin dye and just flood the entire slide like we did with the crystal violet originally. And as you can see, we now have our flooded slide, and then we're going to go ahead and rinse that with our deionized water, and as you can see, we now have a pink stained gram-negative and a purple stained gram-positive, meaning our experiment was a success. Now all that's left is we just need to view under the microscope our results just to verify that there's no misinterpretation of our results. We can see that our gram-positive is stained purple and our gram-negative is stained pink, and that our unknown has tested gram-negative because it is stained pink. Thank you everyone for watching, and I hope I helped you to remember the steps of the gram staining technique.